Welcome to Jinnious Lab. Today we will cover IB Biology B2.1.1-2, 8 to 10, Fluid Mosaic Model.
In B.2.1.1-2 and 8 to 10, we'll be looking at fluid mosaic model. So fluid mosaic model shows how the cell membranes are composed of phospholipid bilayer. And phospholipids are the primary molecules in the cell membranes, and they are composed of a hydrophilic head and a hydrophobic tail, as shown in the picture on the right. So in their hydrophilic head, they have a phosphate ion with a charge, which makes the head polar and hydrophilic. And in the hydrophobic tail, they have fatty acids, which are nonpolar and hydrophobic. So essentially, they are amphipathic, having both hydrophilic and hydrophobic properties. So in water, these amphipathic molecules naturally align into a bilayer with the hydrophobic tails facing the inwards, and then hydrophilic heads facing the watery environments on the outwards. Then we can move on to the selectivity of the cell membrane and how this selectivity is formed by the unique structure of phospholipid bilayer. So the permeability to the membrane of a substance really depends on the polarity and size of the molecule. And because we have the long polar hydrocarbon tails of the phospholipid, which forms the inner part of the membrane, lipids bilayer will become selectively permeable to certain molecules. So large and polar molecules cannot directly diffuse across the membrane because we have a very long fatty acid bilayer, bilayer composed of long fatty acid tails to go through. So for example, we have ions, which are charged and hence hydrophilic, glucose, and amino acid, which cannot directly diffuse across the membrane. However, on the other hand, small or non-polar, hence hydrophobic molecules can freely diffuse across the membrane because they are hydrophobic and hence lipophilic. They can pass through the largely lipid bilayer relatively easily. So an example is oxygen, and oxygen, due to its small size, can pass through the lipid bilayer directly. And then we can look at the intermembrane proteins, which are embedded into our cell membrane. So intermembrane proteins have many functions, and the main ones include intercellular joining, enzymatic activity, transport, cell-to-cell recognition, and anchorage, attachment, and signal transduction as well. So when they act as transporters, they usually facilitate the movement of substances across the membrane. Previously, we looked at how the membrane was very selective, so it sometimes allows the transport of molecules that cannot directly diffuse across the membrane. And then we also have receptors, which allow cells to communicate with the external environment by binding to substances such as hormones. And we have enzymes, which will catalyze certain reactions. And then also they help maintain the cell's shape and structure. And then some of them have a role in signal transduction, where they transmit signals within the cell. Also, they serve as an attachment point for the cytoskeleton and extracellular matrix. So intermembrane proteins can be actually classified into two main types with regards to their orientation in the membrane. So there are intermembrane proteins which are embedded in one or both of the lipid bilayers of the membrane, which are called the integral proteins. So you can see in the picture on the left how integral proteins largely occupy the membrane. And then we have peripheral proteins, which are attached to one or other surface of the bilayer. And then these proteins do not only differ in their orientation, but also slightly in their function. So for the integral proteins, because they are embedded within the lipid bilayer and extends across the entire membrane, they have unique functions. So they often form channels or pores within the membrane, allowing the substances to pass through. And they have other functions such as transporters and receptors and enzymes, like we discussed before. We also have peripheral proteins, which are loosely attached to the outer or inner surface of the membrane, as we discussed before. And these proteins often have functional domains that interact with other molecules. So they become the key player in cellular signaling, usually. So because from this unique property, they have functions that include cell shape and structure, where they maintain integrity of the cell, and they also transmit signals within the cell, and they often serve as the attachment points. Now we can move on to the modified protein and lipid in phospholipid bilayer. And here is the diagram of the cell membrane model. And we will be mainly be focusing on the glycoprotein and then glycolipid over here. So glycolipid and glycoprotein can be formed by glycosylation process. And this means the attachment of carbohydrate chains into the membrane molecules. So glycolipids refer to the carbohydrate chains that are attached to the phospholipids. And then glycoproteins refer to the carbohydrate chains that are attached to the membrane proteins. So glycolipids are located on the extracellular side of the cell membrane, and they have variety of functions, but their main ones are cell adhesion, cell recognition, and maintaining structural integrity. So oftentimes they facilitate the binding of a cell to the extracellular matrix or to the other cells. And an example would be the sperm binding to an egg. They also are involved in cell-to-cell communication and signaling, often acting as receptors for hormones and other signaling molecules. So for instance, our ABO blood group antigens are actually glycolipids. And then with regards to the structural integrity, they help really in maintaining structural integrity of the matrix by linking the extracellular molecules and contributing to the cohesiveness of the matrix. Now we will be looking at actually drawing the fluid mosaic model, which you are required to know. And then when you draw to the diagram of fluid mosaic model, you are required to include certain structures. First of all, you need to include phospholipid bilayer with correct orientation and show that they have very distinct polar and non-polar regions like the hydrophilic head and hydrophobic fatty acid tail. And then we need to show that there are integral proteins which are shown to be spanning or partially embedded in the bilayer, and peripheral proteins, which should be shown as a smaller protein that are anchored to one side of the membrane. And you have to also draw glycoproteins and glycolipids, which are oriented toward the extracellular space. And then also cholesterol, which can be illustrated between the phospholipid tails, primarily in animal cell membranes. So again, these are the components of fluid mosaic model that you need to include organized in a chart.
For IB45 Genius, Jinnious Lab, don't forget to subscribe and like.